Okay, thank you so much, guys, for coming. So we're already on material for midterm 3, and we're going to start off with lecture 11. We're going to talk about the nervous system. We'll just start talking off about the cells that are found in there, the neurons and the glial cells, and the type of neurons they are. We'll talk about multipolar, unipolar, and bipolar neurons, and then we'll talk a bit about the functions, of course, of the nervous system, and talk about the characteristics of neurons and functions of neurons, because there's something very different about neurons, when you compare them with other cells in the body, because they don't have centrioles, they are not able to regenerate. Okay, so it's very important. So the number of brain cells you have, neurons, in fact, it goes down as you get older. You need to use your brain in order to maintain your neurons because they don't regenerate. What they do is just repair damage to parts of the cell. And so they have a very complicated endoplasmic reticulum system that has got ribosomes on them called mesopods, and we'll talk about that because they do not regenerate. So of course we know about it. It's the master control of the whole body. It's like the hard drive of a computer, really. It controls everything that happens in the body. All the homeostasis we talked about in the beginning is controlled by the brain, and also the endocrine system follows second with the hormones in terms of controlling the functions of the body. So, of course, we know about sensory input, which is gathering information from the internal environment and the external environment, and conducting it to the central nervous system, where it is processed. So the central nervous system function is integration, making sense of the sensory information that's going up here to the brain. Okay, so that's the sensory part, and then the integration part, because it's not good receiving information in the brain and not being able to make sense of it. Okay, you gotta be able to decode the information and then act upon it. You gotta be able to respond. In so far as controlling the muscles, to move, for instance, maybe you may be touching something hot, like I did in the lab the other day, and I moved my hand off very fast in order to limit tissue damage. Or it could be a gland that's releasing some hormone, the thyroid gland. Maybe you've taken some milk and you need to absorb the calcium, and then the calcium is taken in by calcitonin and so forth. So control of the muscles and the glands is the response that comes after you get sensory input, or when there's a change or a stimuli. All right, and then of course all the homeostatic mechanisms are controlled by the brain. Remember when we talked about the components of a homeostatic mechanism, and we said you need a receptor, right, to pick the information up. Then you need a neuron, sensory neuron, to take the information up, and then the brain will make sense of it, the control center, and then we will send a response or instructions for the effectors to take action in order to maintain homeostasis, which is a relatively constant internal environment. Right? And then, of course, mental activities. It's the seat of intelligence. All right? So, of course, you got to be able to tell me the functions of any system that we talk about. Okay? It's a given, right? So, we get two major parts of the nervous system, right? The central nervous system includes what and what? The brain and the spinal cord, right? Mm-hmm. And then the peripheral nervous system, which includes what and what?
Receptors and nerves.
Yeah, receptors and nerves, which include what and what? We got spinal nerves and cranial nerves. Yes, that's all periphery. Periphery is the perimeter, is on the outside. So anything that goes off the spinal cord and the brain is on the periphery. All right? So we will talk today pretty much about how this whole thing is divided up. So we got the nervous system here, and then it's divided into the central nervous system, and then also the peripheral nervous system. All right? And then the peripheral nervous system is divided into the sensory nervous system, all right, and also the motor. So today we're going to kind of talk about the organization of the nervous system. All right, which itself is divided into the somatic. Somatic, remember we talked about how muscles arise from somites. So that's why the nervous system branch that controls muscles is called the somatic nervous system. All right, and then anything that's automatically controlled in the body is the autonomic nervous system, right? And the autonomic nervous system has 3 branches, all right? Okay, we get the parasympathetic, which normally does what? Parasympathetic.
Vegetative functions.
Yeah, well, it's normally resting conditions. It normally inhibits. So whenever you've eaten a McDonald's meal, or not that you should go to McDonald's, okay? It's the most popular restaurant. Or when you're eating a meal, when you're resting, that's when you are doing what? You're digesting, and the parasympathetic system is going to be active via the vagus nerve. All right? And then sympathetic usually increases, right? And I'll show you these branches in a bit. And it's the fight situation kind of deal. The epinephrine, no epinephrine. So it sends you into motion, into doing something. And then because the digestive activities have a very complex network. In fact, there's some sort of neurons right there in your digestive system, which we will cover when we do AMP2. There's the enteric nervous system, which really deals with digestion. Okay. So one of the things I do in the exam is I ask you to show me how the nervous system is organized as a whole. All right, and you show me all these divisions, and show me what is divided up into what, or what is comprised of what, like the CNS being comprised of the brain and the spinal cord, and the peripheral nervous system being the receptors, spinal nerves, and cranial nerves. Cranial nerves is only 12, and there's 31 pairs of spinal nerves on either side of the spinal cord. All right? So these are the parts of the nervous system. The central, we talked about it, is the brain, and the spinal cord is the central nervous system. You get reflexes originating from the brain, from the brainstem. You also get a lot of reflexes originating from the spine. So the next lecture we're going to talk about spine and plexus. We'll talk about these reflexes. All right? So today we're just going to talk about how the system is organized, of course. We just did the skeletal system, and we were talking about the cranium, which protects what? The brain. Okay? So it's encased in that cranium, the brain is. And what encases the spinal cord? Yep, the vertebral column encases the spinal cord. All right. And so we all know about that. And then, of course, the peripheral nervous system is everything on the periphery. All right. Okay. And consists of any nervous tissue outside of the central nervous system, and it's got, of course, sensory nerves and the receptors. Sensory receptors and nerves, of which there's cranial and spinal, right? So the 2 anatomical divisions, which are the major 2 divisions, will be the peripheral nervous system and the central nervous system. Okay. It is the peripheral nervous system that's divided up into all those parts that I just talked about, which I'll show you again in a little bit. Okay. So peripheral nervous system, because of the receptors and the brain nerve endings, detect stimuli in the environment, and it will send it to the central nervous system for processing. So the nervous system, the central nervous system, will integrate it, make sense of it, and perceive the information, and then act upon it or not. Because it is not all the time that the brain, the central nervous system, has to respond where it gets stimuli. Okay, because homeostasis is maintained within a certain range, right? It's not like doesn't flatline. Okay, so it's maintained within a range. So if the variable falls within that range, the body doesn't have to do anything; it just stays. But if it falls out of the range, that's when the body has to do something. Okay. All right. So we got the two divisions of the peripheral nervous system. Is the sensory, okay, which takes information up to the brain and the spinal cord, and then we get the motor. It's the motor that's divided up, okay? Because with this, not only do I want you to know the parts, I want you to know which part is divided into what, okay? So when I ask you to show me how the nervous system is organized, that's what you do. You show me with arrows, kind of like an organization chart, if you will, and show me what is divided into what, because I usually kind of like that question for the exam. With a whole bunch of 5 points. Okay, so okay, the motor division is divided into the somatic, which we said controls muscles. Somites are the structures in the fetus that will eventually become muscles, hence the name somatic. Controls skeletal muscles, okay, for movement and so forth. And remember, this is voluntarily controlled, except a few instances when we have reflexes. Okay, and then the autonomic, of course, anything automatically controlled, like digestive activities, blood pressure, and all those things. Okay, with a smooth muscle, and so forth. Okay, so the sympathetic fight or flight, you know that because sympathetic. If you're sympathetic to somebody, you kind of give them something, you kind of add to them. Okay, so this one is one that stimulates. Parasympathetic, you kind of against, so you know it will inhibit. So you can tell those systems apart. Okay. Because I want you to be able to describe these branches, kind of like give you a little function of it, okay? Parasympathetic, of course, regulates resting functions and digestion. And digestive activity is so elaborate that it has its own system called the enteric, okay? And that controls all the digestive system activities, all right? So tell me about the organization. Tell me about these parts and what they do of this nervous system. Okay, it won't be enough to just say, okay, sensory division is sits there. Okay, you get half a point for that. Okay, so you gotta tell me sensory takes information up to the central nervous system. All right, so these are them. Okay, and we've kind of done this ad nauseum, so you know you need to know this, right? Because this is the basic part of the nervous system and how it's arranged. And then after this, we know how it's organized and what the functions of the parts are. We're going to talk about the different cells and what those cells do, okay? We're going to talk about the different types of neurons. So we know all about this, right? We know all about this chart, and we know what each of those parts is doing, all right? We covered it. So to give you an example, this is like a homeostatic mechanism, right? Pretty much, considering those 5 steps which was in the exam last time, okay? So whenever there's an input or a change or a stimuli is the same thing. A stimulus is some change in the environment. So what change has happened in his environment? What change has happened? What's it?
He feels threatened.
Yeah, you feel threatened by what? So what's the change? The change is the appearance, the stimuli, in this case, is the appearance of a bear, right? That's what it is. So that's the change. And what part of his body is going to sense that change? When a bear comes by, what do you do to it? You see it, right? So the eyes are going to sense that. And then those neurons, the sensory neurons in the eyes are going to send the information to the brain and say, you know what, there's a bear over there. You know what I'm saying? So that's the sensory division doing that, sending it to the brain. And then the brain is going to say, oh, bear, bad. It's going to do something. It's going to send information now to the muscles, to the skeletal muscles in this case, because you're going to need to do what? To get out of there. Okay? So that's the response, right? We're on the response side, and these are motor neurons, okay? So these are motor neurons. Motor because it causes you to do something. And on this side, we get sensory neurons, right? Because they're sending information to the brain. And over here, we've got a receptor, right? A sensory receptor. That is gathering that information, right? And then you need to send information to all your branches of this autonomic system, you know what I'm saying? Because not only do you want to send more blood to your muscles to get the hell out of there, right? You're going to want to stop all those resting activities in your parasympathetic side of things or your enteric system, because you don't want to be digesting your burger while you're running away from, you know what I'm saying? So you stop activity there by, stopping here, and here it's a check, okay? And the response is you are running there like, boat to get out of there, okay? So that's an example of a perfect homeostatic mechanism. It could be an external stimuli, or it could be an internal stimuli. Or say calcium levels fall down in your blood, then you release certain hormones, and then they will be able to break that calcium hormone and whatnot. Okay, so but this is a good example because it causes emotion, right? About the brain. All right, so now we talk about the cells, the different cells in the nervous system. So there's pretty much two basic cells: the neurons, which are the ones that pretty much set the number of, okay, because they do not divide. And we know that neurons are excitable, right? That's why they're able to conduct an action potential on the nerve. One thing about neurons is that they have processes coming out of the cell body, which are kind of like extensions of the cytoplasm. You know what I'm saying? Okay, so I'm going to draw me a neuron. We kind of saw this. Did we do the neuromuscular junction? Yeah, we kind of saw this, and I kind of drew this. So this is the cell body, okay, where you will have all those organelles, the nucleus and whatnot, okay? And then what did I say about the neurons? What do? They branch profusely, right? You can get up to 10,000 of these branches on just one neuron. Yeah, on one. Mm-hmm. Imagine if it's controlling your thigh muscle. How many cells do you have in your thigh muscles? Like a whole bunch. That you couldn't possibly have one neuron going to each muscle cell. Couldn't. So it's more than 10,000. It can go up to 10,000 branches in just one. Okay? So these little processes, the shorter ones, are called dendrites. Okay? And I'll show you the structure. And these conduct, they're like sensory receptors, and these are spread all over to gather information from all the surrounding. And it kind of increases the surface area of that, right? Of that cell. Okay, so dendrites are transmitting information towards the cell body. Okay? All right. And then all that information that has been passed by the dendrites to the cell body is gathered at a certain specific part here called the axon hillock, or sometimes called the trigger zone. Okay? Because all that information is kind of sort of united here, and then it's going to go to the axon, okay, which is conducting impulses away from the cell body. It's going to wherever it's going now, either to the brain if it's a sensory one, okay, and it's going to tell the brain, okay, this is what's happening, or it's going to the muscle, and then it's going to end up at the neuromuscular junction and tell it to contract after release of acetylcholine, right? Okay, so this is the basic structure, and this kind of neuron that has a whole bunch of these processes, a bunch of dendrites, and one axon, this is what's called a multipolar, and most of them are like that, okay? But then we'll talk about the other two that have two processes called bipolar, and one that is one process called unipolar, okay? All right, but I just wanted to kind of sort of show you the basic structure of the neurons, all right? And these neurons, of course, like I say, they don't divide because they don't have centrioles. So they have a whole bunch of support cells, which makes the bulk of the brain actually, called neuron glial cells. These are non-excitable cells that usually surround neurons to support them and to put them close to blood supply for nutrients and oxygen and so forth, okay? So they kind of sort of support and nourish neurons. Okay, so that's their function. So if I ask about the two basic cell types in the nervous system, you're going to talk about the neurons, and you're going to talk about them being the excitable ones that are able to respond to stimuli and send messages, okay? And then you talk about glial cells. They are called Neurons Glial cells. For short, we just call them glial cells, and in the exam you're allowed to use glial cells, okay? I want to say more abbreviation to that, okay? So call them glial cells, and these are kind of support cells. And the bulk of the brain is actually, see what I'm saying? Over half of the brain's weight are neuroglial cells. See what I'm saying? Okay. So those play an important role in order to maintain our neurons. Since they're not going to be dividing and replenishing, we need to take care of those. Kind of take care of those stuff. So and about less than 20% is extracellular space. So it's kind of packed in there. In fact, there's a part of the brain that's called the cortex. It's about 5 millimeters thick, where the majority of the neurons in the brain are actually packed, maybe 75%, 3/4 of it. And so it's densely packed in there, okay? And it's got all the cell bodies, okay, in there. And it's usually called the gray matter because cell bodies are not myelinated. Myelination is when they are surrounded by these cells, okay, the membrane, of those cells, which is whitish in appearance. And usually it only occurs in the axon, and that those, like this axon, will have this myelin sheath, and the myelin sheath goes in breaks, and what it does is that it shortens the impulse so that the impulse, when it's going, it only is conducted in these parts that are not covered by the myelin sheath, because the myelin sheath insulates. We'll talk about that in a little bit, but okay. So they're kind of densely packed in there in the brain. All right. So the basic two cells, neurons and glial cells. So we'll talk about neurons first because they're very important, and like I say, they need to be taken care of really well because they do not divide. Okay. So these ones receive the stimuli, okay, via those dendrites. See, the dendrites pass information to the cell body, and then transmit action potentials via the axon, which is very long. It could be an axon could be a few millimeters or 1 meter long. Yeah, imagine a motor neuron starting in your brain, okay, and ending in your big toe. Right? That's long, especially if you are, shark, which is tall. So it's like a couple of meters or something. You know what I'm saying? So yeah, so it can be that long. An axon can be that long. In fact, when we divide, when we do the brain, when we do the muscles, I'll be showing you a whole bunch of nerves, bundles of axons, and you can see them because they are really thick bundles, like that. Okay, so what are the components of a neuron? We talked about the cell body already, okay, and then we talked about those short processes that surround the cell body called dendrites, all right, and then we talked about one axon. There's only one axon, okay. Very few times the axon will branch, and it will have branches at right angles to the axon called collateral branches, okay. But most of the time it's just the one axon and that's it, okay. So know the components of the cell body is also called soma, okay. And it's got the organelles in it, with nucleus and whatnot, and those nasal bodies which we're going to talk about. Nasal body is a whole bunch of endoplasmic reticulum, rough endoplasmic reticulum, and of course the axon. These are all extensions of the cytoplasm. So that membrane will continue and will transmit or propagate the action potential along it until it reaches the destination, which is the axon terminal, which is branched many times. Okay, so gotta know the parts of the neuron. Okay, what the function of those parts of the neuron are. Okay, gotta know the two cells, the neurons and glial cells, right? The two major cell types in the nervous system, and then of course how it's arranged, how it's all divided up, and what the functions of those parts are. Okay, starting from the nervous system all the way to the enteric nervous system that controls the standard of what the digestive activity. Okay, so we get you see all these organelles in there. It's really packed, rich with all these organelles. You see, you got your rough endoplasmic reticulum, and then you get the nasal substance that we were talking about that helps to form a protein, so that you can prepare, let's say, a dendrite breaks off or something like that. You replace that by creating all the proteins and whatnot that's required, right? And then, of course, this part is the cell body, and these are the dendrites, all right? And some mitochondria in there, of course. And then, of course, the axon hillock, which is kind of at the base, or sometimes called the trigger zone. So the trigger zone or axon hillock is the same thing. So this is the myelin sheath I was talking about. So the axons can either be myelinated or not. Okay. So the ones that are not myelinated will be part of the gray matter because they're not white. So only the myelinated axons will be part of the white matter. Because at the end, I'll talk about gray matter and what makes up gray matter and white matter and what makes up white matter. The only thing that can make up white matter in the brain is those myelinated axons, okay? Because those are the only pathway that gets myelinated. Dendrites don't get myelinated, they're way too short, okay? And the myelination, the whole point about it is to insulate that part of the axon so that it kind of doesn't conduct. And so the conduction will only occur in these spaces, the small spaces, so that the impulse jumps there, and that's called saltatory conduction. And those little parts that are not covered are called nodes of Ranvier, okay? Right? Nodes of Ranvier. And so the impulse will, it will really speed it up from about 1 meter per second to about 150 meters per second in speed. Unmyelinated and myelinated, okay? That's 150 times faster. Okay, you see, this is one of the few times you get a collateral branch at right angles to the axon. Okay, so that's a collateral axon. So a few times that happens, but for the most part, it's just the one axon. Okay, and that's that.
And when would that happen? When would you have
a collateral? Maybe when it needs to send the impulse to some other part. It's also another way to multiply the neuron's ability, because you can have only so many, like I said, up to 10,000 terminal branches. Okay, so another way, if this neuron is kind of controlling a whole bunch of stuff, it may have a collateral branch, so that the collateral branch will also have terminals that can split. See what I'm saying? So it kind of increases, if it's kind of taking care of the wide. I would say, for instance, the neurons going to my thigh muscles, because these are big muscles, you know what I'm saying, they may have collateral branches, you know what I'm saying, so that they can control more. Rather than just profusely branch out, it can get a little bit complicated there. So you may have collateral branches depending on the complexity of the part that's being controlled by that neuron. Okay, so that's a good one. That's a good question. All right. So that's a multipolar, by the way, an example of a multipolar neuron, because it's got many processes, many dendrites, one axon. Okay. So we talk more about the neuron because again it's very important. Why? Because it doesn't divide. So we need to really take good care of it. So what does it contain? Of course, a nucleus and a nucleolus, so that it can direct all the activity of the cell, make new proteins and so forth, right? So what I was talking about, those nasal substance, aggregate of rough endoplasmic reticulum. From last time, what does the rough endoplasmic reticulum do? Yeah, protein synthesis. So you're going to be making a whole bunch of protein. So there's so much of this rough endoplasmic reticulum, we call it nasal bodies. Okay? Because, and a bunch of free ribosomes. You know what I'm saying? Free ribosomes, we said they make what? What kind of proteins do free ribosomes make? Remember we said free ribosomes is an organelle, and what's the function of free? And it goes in the exam too. It was on your last exam. They make proteins that do what? What happens to those proteins? They're already in the cytoplasm, so they're going to be used by the cell itself. Okay? So that's why you have a whole bunch of these free ribosomes, because the neuron itself is going to be using those proteins to repair itself, because it doesn't divide. Right? So it makes sense, right? You see what I'm saying? Makes sense? primary site of protein synthesis, of course. And those free radicals are making a whole bunch of proteins that are going to be repairing all those dendrites when they fall off into whatnot. Okay? Repairing all those myelin sheath and some of those proteins and whatnot. Okay? And then it's got Golgi apparatus, of course. You know why? Because the Golgi apparatus is the one that kind of packages all these proteins, kind of modifies the proteins, gives it this tertiary structure, like its final folding and so forth and whatnot. If it's a glycoprotein, the protein and the carbohydrate are joined in the Golgi apparatus, right? So it gives it its final structure, the protein. So a bunch of that, you need the energy, all right? And other organelles are present, like vesicles and whatnot, and all those other things. So the cell is really paired with the full complement of organelles. Okay? We've talked about other cells, like muscles, that don't have a whole bunch of organelles in them because all they're doing is contracting. Okay, so that's why at the beginning we learn about cells, the basic structure of cells, and what the function of these organelles are. Okay? So when we proceed, you're not really learning anything very new here. Okay? You're just applying those same things to whatever specialized function for that cell. You see what I'm saying? So that's why the subject, you kind of have to keep up with the information and you are adding on. It's not like you finish with midterm 2 so you forget. No, you don't. You gotta keep that information because it's building up on top and you are learning new things that depend on your understanding of what you have just done. Okay? And the way it's structured is such that you learn it in steps like that and it builds up. Okay, so like I said, it's got no centrioles. Okay, centrioles, what do centrioles do? What do they do? Remember when you did mitosis in biology, what do centrioles do? Okay. Centrioles, they move the DNA. You know how when the DNA goes into the, after it becomes the chromosomes and all those chromosomes like that, those put in the center of the cell like that, right?
Venus says they do a dance. Yeah.
Okay.
Yeah, that's what she said. She said they do a dance.
Okay, they do a dance. You know, they'll be pretty much, they are like rod-shaped proteins that will move these chromosomes onto the center, so that when it splits, it will split. This one, this chromosome goes here, that chromosome goes there, and then it splits when the cell is dividing. Okay, so if a cell doesn't have centrioles, it's not going to be able to do that. So it's not going to be able to divide. See what I'm saying? Okay, because you know the thing about it is that if I was to ask about all these things about neurons, if I say to you they don't have centrioles, and you don't know what centrioles do, then you won't even be able to give me the whole answer. That's why I'm going through that. Okay? I'm assuming you should know what centrioles are already because you've kind of done mitosis and all those things. See what I'm saying? So if you know those things, it makes it easier for you to remember the information. This is why I'm going through this. Okay? Well, you know, if it doesn't have centrioles, then boom, oh, the cell cannot divide immediately without even thinking about it. You see what I'm saying? So that's how you, because I'm going to want you to say, okay, it doesn't have centrioles, so what? So it is not going to be able to. Amitotic means it can't do mitosis, so it can't divide. So that's why you need to go through all these lengths of having a whole bunch of meso bodies, whatnot, to make proteins to repair. Clusters of cell bodies, of course, when they are in the central nervous system, they're called nuclei. When they are in the peripheral nervous system, they're called ganglia. That's it. Okay? So just know that. And this is showing you the parts of a neuron. I'd like you to kind of look at these diagrams later on. You see the hillock, which is the base of the axon, and then the axon and so forth, and then the cell body, nucleus, dendrites, and on all that. You see the nasal substance over there? Almost the whole cell is made up of nasal substance, which is pretty much those free ribosomes and which part? Endoplasmic reticulum or Golgi body? What is nasal substance made up of? Raphael. Sometimes you get a multiple choice question like that. That will list all those organelles. So, like we said again, this is pretty much repetitive. The part of the axon that gathers the information from the dendrites and then conducts is the axon hillock. Okay, is the trigger zone. Okay, you could call it hillock or trigger zone. I don't really care. Okay, and then of course the axon is a slender process. That I said is can be a few millimeters. Can be a meter long, depending on how tall you are. Because some of them may need to go all the way down to the big toe. from the brain. So that's the whole length of your body, right? So usually it's unbranched, of course, like you say. Okay. So you could say a lot about a neuron, right? You could say a whole bunch. I could ask a 10-point question, just asking you telling me about the neuron and the parts that it has and what they do. Okay. Okay. So it's very rare that it has these branches, like I was saying, the collateral branches, the ones that appear at right angle to the axon. Okay, very few times, but it does. Okay, presynaptic terminal, you see what I'm saying? Spring, you see this? A whole bunch of branches, you know what I'm saying? A lot. 10,000 is a lot if you was to count to. And that's one axon and it's branching like that. So you can imagine that. I mean, you know, that reduces the number of neurons you need. Because if you were to have a neuron for each cell, for each gland, get by. It's just going to be too much wiring. It's going to be so confusing and whatnot. It just doesn't work. And then we know about the synapse, right? We just did that. We know about the synapse. What is the synapse?
Between nerve cells.
And what about that junction? It can be between a neuron and a neuron. It can be between a neuron and a muscle cell. And if it's between a neuron and a muscle cell, it's called the neuromuscular junction, right? So what did we say synapses? What is it? Can you describe synapse? What is it? Is it a physical connection or is it a functional connection? Yeah, so that means it's going to be what kind of connection? Functional. It's going to be a functional connection between either a neuron and a neuron or a neuron and a muscle cell. You know what I'm saying? Because they don't touch. They don't. You said there's a gap, so there's not a physical connection. So there's a functional connection because the neuron is able to affect the next neuron or is able to affect the next cell, which could be a muscle cell or whatever. Okay, so synapse is a junction between nerve cell and it's a functional connection. Okay, so whenever I ask you to define synapse, I want you to say functional connection. Okay, because there's not a physical connection between those two, but there is a functional connection in that they're able to affect each other, but they don't touch. They are not connected. All right. So if we have bundles of axons in the nervous system, they're called tracts. Okay? Outside of the nervous system, they're called nerves. And I said I was going to show you, remember when you do the muscular system, I was going to show you a whole bunch of nerves. When we separate the muscles out, you can't miss them. The nerves are so big, the bundles of axons in the cat. You'll be able to see them, okay? So, you got to know that tracts in the CNS, and then if I say what are tracts, you'll be able to tell me, right? You'll be able to say, oh, these are, axon bundles in the nervous system, in the central nervous system. And they form part of the white matter too, okay? If they're myelinated. All right? And then nerves in the periphery, like the spinal nerves, like the cranial nerves. All right? So now we talk about the shapes and, of these neurons. We've just described the multipolar, remember? One with a bunch of dendrites to the cell body and one axon. We just did that. So that's a multipolar, many, polar, many branches, okay? So there's a bunch of dendrites, only one axon. Okay? So one axon is always one axon, okay? Because it's one that finally conducts all the information to the brain or to the muscle if it's a motor. Okay? So all the interneurons, neurons that connect other neurons are going to be multipolar, okay? Because they're going to be pretty much connected to a whole bunch of other neurons. Okay? And they're going to be needing to gather information from a whole bunch of other neurons. So they have a bunch of dendrites, of course. And all the motor neurons are like that. Remember motor neurons? They'll be controlling a whole bunch of these muscle together. Many dendrites, one axon, which will branch a lot. All right? And then the bipolar, just like the name says, you know what I'm saying? It's got two. It's got two. So you got your cell body there, and you got one branch that acts as a dendrite, one branch that acts as a axon. Exactly. And most of the special senses are like that, like in the eyes and whatnot. They have bipolar. Those are special senses. So it's normally, reserved for special senses. Okay? And most of the other ones are in the motor, division. Motor neurons, whatnot, are multipolar. Okay? And we know what the shape of a multipolar is, right? Okay, so that's bipolar. And then unipolar, you guessed it. It's got one axon, really, one branch. And I'll show you nice pictures later on. So this is what it is, you know what I'm saying? So it's got a part that kind of acts like a dendrite, and it's got a part that kind of conducts to the, okay, sometimes they call this part the peripheral branch, you know, because it's sensing from the periphery, and, you know, okay, peripheral, from peripheral nervous system, right? And then central branch, it's really one axon, you know what I'm saying? It's one branch. You see what I'm saying? How it does, there's a small gap between, you know, like a connector between the cell body and the one axon. And then there's a central, the one that is conducting, the part of the axon that's conducting to the central nervous system, that's the central branch. Okay, so sometimes they call it that. And again, this is special senses, you know, things like that, sight, hearing, whatnot, together with bipolar. Okay, so these are mostly sensory. Okay, and for special senses like sight, hearing, taste, whatnot, and also balance is a special sense, and it's by the ear too. Okay, the ear takes care of that. So that I'd be able to tell me about, of course, the two basic cells, the organization of the nervous system, all right, and then the neurons, the characteristics, functions, and the different parts and the functions, and then the different kinds of neurons, you know what I'm saying? Look at the multipolar, unipolar, bipolar, okay? And be able to describe them for me, okay? In the exam, don't just draw it like this and then stop there, okay? Because I become blind at that time. I want you to be able to tell me in words what it looks like, okay? And then if you want to draw it, you can draw it. You can draw it and then describe what you just drew if you want in words. So again, multipolar here, and then bipolar, and then unipolar. So this is what I'm saying is sometimes called the peripheral branch, okay? And this is sometimes called the central branch, because it's one that conducts information to the central nervous system, right? Okay, so know those three different types of neurons, and kind of where you're likely to find them, okay? Multipolar, most of the interneurons and motor neurons are like that, multipolar. And then special sense, sight, you get a bunch of bipolar there. Smell, you get a bunch of bipolar neurons, okay? And unipolar as well, special senses. So in the exam, I might say, okay, can you ask me, this will be like a 2-point question with a 3B, right? You know, so you get 3 points for one for bonus. Ask about the types of neurons, okay? Name and describe them, and give me like a location or function where you're likely to find them. Then you talk about that, okay? Multipolar, interneurons, motor neurons, bipolar, special senses, sight, smell, hearing, and stuff like that, together with the unipolar, okay? And then describe the processes and so forth. Okay, so that's a 3-point question. It's going to come in the exam. Glial cells. Now, we said a whole bunch of the weight of the brain is going to be glial cells, right? Because these need a lot of support. They need of TLC, right? The neurons, because they're not going to divide. So you need to take good care of them. And so we got a whole bunch of these glial cells, of which there's 2 in the peripheral nervous system and 4 in the central nervous system. And we'll talk about the different 4 types in what they do and their characteristics in the nervous system, and we'll talk about the 2 in the peripheral and what they do, okay? So, clear? They provide support, of course, to those neurons that we say are very delicate. They do not divide, so we need to take good care of them, right? And then segregate and insulate. Again, what are these neurons doing? What is the basic deal about these neurons? They conduct electrical impulses, really, which are called action potentials. So imagine if they were going to be cross-contaminating everything. Then you won't be able to pinpoint something and stimulate only that. You stimulate the whole body, like doing this. So yeah, seriously. So you need to insulate them. So they insulate them, and we'll talk about some of the glial cells, you know, in the periphery that do that. That will insulate it so that it's like the electrical wire. Have you seen electrical wires that are bare? They always have a rubber covering on it to insulate it. Otherwise, your whole house will be like that, with all the lights on or whatnot. Okay, because everything will stimulate each other. Okay, so you need that And they also, when the young neurons are kind of growing and growing in length, they're guided by these, glial cells, and so that they have the proper connection, so that it grows into the right spot. If it's going to control your big toe, it will grow right there. This is usually happening in a young, or when the body is forming in utero, right? And these are growing, so they're guided by that. Okay. And as they are guided, they are also surrounded. As it's growing, and it's surrounded already. It's like building it and surrounding it so that it's all insulated. And then also promote health and growth, of course, of those, neurons. Because we need them. So sometimes I might say, what are the basic functions of these glial cells, that do the support? You can talk about all this stuff here. Insulating them, supporting them, providing all these nutrients, guiding the young ones so they grow to the right spot and stimulate the right part of the body. And this pretty much promote the health of these units. So there's going to be 4 glial cells in the central nervous system. So oligodendrocytes, microglia, ependymal cells, and astrocytes. Astrocytes look like a star. The astra is star, right? So you can imagine it looks like a multipolar. You know what I'm saying? Microglia, they are what? Just by the name of it. Micro?
Small.
Small. They're small glial cells with less dendrites, less processes, and small. And these are the macrophages of the brain. Okay. What do macrophages do? They engulf what? What do they engulf? What are macrophages? No? No, they're not dead cells. They're so much alike, actually. They protect you. Macrophages do what? They engulf what? Macrophages engulf what? What other cells? What kind of other cells? Bacteria. Who said that? Did you say that? Yeah, bacteria. So these ones, microglia, are the bacterial. They engulf bacteria in there. Macrophages do that. They engulf bacteria. So microglial cells engulf bacteria in the brain. You don't want, bacteria up here. And then ependymal cells are involved in making cerebrospinal fluid, the one that cushions the brain, and the one that has all these ions and so forth. So that, because action potentials are movement of ions across the membrane. So cerebrospinal fluid has got a whole bunch of these ions and it's kind of, breathes all these cells. Okay, so ependymal cells do that. Okay, oligodendrocytes, they are few dendrites. Okay, and these ones, they create the myelin sheath in the central nervous system. The myelin sheath that we talked about that surrounds those axons. Okay, so they create the myelin sheath. So we'll talk about that. Astrocytes have a whole bunch of functions. They involved with the blood-brain barrier, a lot of stuff they do, repairing and whatnot. So they do a bunch. So we'll talk about that. Okay, and then glial cells in the peripheral nervous system, we pretty much have Schwann cells that do the same function as oligodendrocytes. Okay, which I said is what? Yeah, produces the myelin sheath in the periphery. Okay, and satellite cells kind of do some support. Satellite cells do that in the peripheral nervous system. So we'll talk about each one of those. You know what I'm saying? And then so we start with the astrocytes because these are the busiest. Okay, so the most abundant, the most versatile, highly branched. Okay, that's why they're astrocytes. Astra is star, so they look like a multipolar, right? Okay, they cling to neurons and their synaptic endings, right? That's what I'm saying, protecting the synaptic end. Because remember, the synaptic end is a little gap between the neuron and that whatever is controlling next, whether it's be another neuron or whether it be another muscle cell. Okay, so it protects that and so forth in the brain. So this will be a neuron actually because it's in the brain, not a muscle cell. Okay, it will be in the brain. So it kind of covers those up. So the basic functions, supports braces those neurons so that they're in the correct position. Okay, and holds on to blood vessels. So it will surround a blood vessel and it will surround a neuron and kind of bring them together. So it will bring it to close proximity to the nutrient supply, the blood vessel. Okay, the neuron does. So it will kind of hold on to it. I'll show you a nice picture of astrocytes. Okay, so anchor neurons to the nutrient supply to the blood vessels and influence functioning of the blood-brain barrier. That's what I was saying. The blood-brain barrier is a barrier that doesn't let anything into the brain. You know, you don't want your control center to be compromised. You know, so the blood-brain barrier pretty much lets in glucose. Very few other substances, very, very few. That's why some of these psychological conditions are hard to treat, because it's hard to find a drug that will enter the brain. You know what I'm saying? So you kind of have to have things that mimic these natural compounds that can go through the. So in the pharmaceutical industry, what they do is they have this kind of process. Well, yeah, you may mimic some parts of glucose in that molecule, but then again, if you mimic too much another molecule, you're going to destroy the function that you want it to do. Because if it looks a hell lot like that molecule, it's going to do the function of that molecule. See, that's where the challenge is. So in the pharmaceutical, it's really exciting when they discovering drugs and so forth. In fact, I used to work on drugs that deal with schizophrenia in the brain. Okay, so they have a process that they use called high-throughput screening. But that's the problem, is that nothing just enters the brain because of the blood-brain barrier, which is the result of these astrocytes, these neuroglial cells. Okay, so there's a little story about that. So this blood-brain barrier is very important. And then, of course, guide migration of young neurons, process substances. You know, like how we talked about how action potential is pretty much ions in, ions out. So sometimes it gets sloppy. You know, ions are left out in there, and then the astrocytes will come and mop up. Just like when you cook in the kitchen, you know how you drop stuff up? So if you have an astrocyte, it will come and sweep your kitchen. Same thing, you know what I'm saying? So you can call your kids astrocytes, you know what I'm saying? Because you tell them to go sweep after you cooked and dropped stuff. So after the axon is contracted and it's leaked all over these neurons, these ions, then the astrocytes come and sweep that up. So mopping and recycling neurotransmitters as well. Because you're not going to be, secreting all these neurotransmitters and then you keep secreting as long as, so they are recycled too. And we talked about those things, events in the neurotransmitter, junction, you recycle that. So astrocytes do that. They help recycling, isolate damaged tissue, limit damage and repair and so forth. So the astrocytes are involved in a bunch of stuff. Sometimes I'll say, can you tell me the function of astrocytes? What do they do? So you list all these things, 5 points. So you see what I was talking about? There is a neuron and these astrocytes, you see it looks like a star. It's grabbing the blood supply and putting it into close proximity to the neuron so that it gets things like oxygen and nutrients and whatnot. So the next set of cells is the ependymal cells. Ependymal cells, like I said, these are columnar cells and they are ciliated, they got cilia on them. Remember those organelles we talked about? Cilia, what do cilia do? They move stuff on the surface, like the cilia in the fallopian tubes. It moves the egg down the tube as it goes towards the uterus, right? Same thing, it circulates the cerebrospinal fluid. So these cells are columnar, they are ciliated. We all know about that when we did the tissues, right? Remember we did, and some of them are squamous. What kind of cell is a squamous cell?
Flat.
Flat, yes, we did that, remember? We did that. So they line ventricles of the brain. Ventricles of the brain do what? They are sort of chambers in the brain that are filled with this fluid, okay? And then also, there are 3 membranes that sort of protect the brain. Very tough one called the dura mater on the outside. And then the arachnoid mater in the middle, and the pia mater, which is right on the brain. Between the arachnoid and the pia mater, there is a fluid. So it's called the subarachnoid space. And the cerebrospinal fluid also circulates in there. So that's what cushions your brain, okay, against your skull. So when you drink, you go to the bathroom a lot, and so you get dehydrated, and then some of that cerebrospinal fluid that's cushioning your brain will start to, to kind of also, drain away a little bit because you don't have fluid. So what happens? Your brain is kind of sort of like really going like this in your head because it's not well cushioned. So that's why when you have a car accident after you've been drinking, you're more likely to have a brain injury because you've been you lose it in the cerebral, the cushion, because when you have a, you know, oh my God, a head-on collision, and you go like this, pretty much your brain and your head are still moving, and the car is stopped, so your brain will slam across on your thingy, on your skull. You get brain injured like that. Okay, because you don't have enough cerebrospinal fluid. So the ependymal cells make that, and of course they're ciliated so that you can circulate the cerebrospinal fluid in the ventricles. And then you got specialized choroid plexuses that are lined with these ependymal cells that are suspended in the ventricles that make up this cerebrospinal fluid. So choroid plexuses are specialized kind of blood vessels looking like that make cerebrospinal fluid. Okay, so help to circulate, and the cilia, like I said, help to circulate the fluid. Okay, so good. So you can see microglia, like I said, these are small and they're fluid dendrites and they promote health. They are phagocytes. They engulf bacteria and foreign bodies and whatnot to protect the brain. Okay, so monitor health of the neurons. So these are microglia. Okay, because normally I ask about these things. And then oligodendrocytes, they form the myelin sheath that will surround the axons in the central nervous system. Okay, you see how the myelin sheath is pretty much that cell. It will wrap itself around, so that the cell membrane will wrap itself in its cytoplasm, wraps itself around the axon, and then that becomes the myelin sheath. So you see the oligodendrocytes, and then it wraps itself around, pretty much around the axon. All right, and the purpose of that is to speed up transmission. Remember, because it will be only conducted, the impulse, on the nodes of Rangia, which are the small spaces that will be left on the thing. Okay, so most of the tracts that connect your left side of the brain to the right side of the brain, the cerebrum, these myelinated axons, and they look very white. You can see them. They are tracts. Remember, tracts and nerve bundles in the central nervous system. Okay, so they're myelinated, so they conduct information fast between the two, the cerebral hemispheres. So Schwann cells, now we move to the periphery, right? We've done the 4 in the central nervous system. Now we're going to the periphery, and Schwann cells, they form the myelin sheath, in the periphery. So they're the same as the oligodendrocytes in the central nervous system that make up myelin sheath as well. Okay, and then of course, especially those 1 meter long axons, you know what I'm saying? They're going to need myelination, don't you think? To conduct the information very fast, to go to your big toe. If you step on a tack, you want to move your foot real quick. You don't want it to take like a few seconds to get up here in a few, you know what I'm saying? So you're going to need myelination, especially in those peripheral. And then of course, satellite cells, we said they support and they kind of sort of nourish these neurons. Okay, so they are around the cell bodies, you know what I'm saying? The satellite cells, so the cell bodies in the peripheral nervous system are called ganglia. So that's where you have a lot of these satellite cells surrounding the cell bodies of the neurons in the peripheral nervous system. So satellite cells, Schwann cells. Okay. So, like we talked about, myelinated axons. Myelination is a way to speed up transmission. Okay. We talked about how the plasma membrane of the cell itself in some cytoplasm will surround and wrap itself around the axon. Okay, so that's what it is. The Schwann cell will be doing that in the periphery, and the oligodendrocytes will be doing that in the central nervous system. All right, and then that part that is covered by the cell and a bit of cytoplasm is called the myelin sheath, and the myelin sheath will insulate, and it's whitish and it's fatty because of that, and it's got protein lipids, and it's segmented, okay, sheath, so that you see there's a part that doesn't have it, and there's a part you see there's the nucleus of the cell, of the Schwann cell, or of the oligodendrocyte if it's in the CNS, right? And it will. So what are the functions? It protects. It also insulates, right? Mm-hmm. It insulates fibers from one another. Remember when I talked about the bundles of axons, which form the nerves. Okay, those will have many different axons of of different neurons. So they have to be insulated against each other. It's just like having electrical wires in a big bundle, but having rubber or whatever surrounding each of those, so that they're insulated against each other, so that they don't stimulate each other. So and it decreases the speed of transmission, because when transmission is going on in there, it will jump to the nodes of Ranvier, which are the parts that are not myelinated, and that kind of transmission is called saltatory. Okay, saltatory conduction. So, and it only happens in the nodes of Ranvier, which are the gaps in the myelin sheath. Saltatory like salt, saltatory conduction. Unmyelinated, of course, axons do not have the actual cell wrapping itself around. But oligodendrocytes and Schwann cells still kind of sort of surround the unmyelinated axons for what? This time for insulation only against each other. Because if you have a bundle of these nerves, you see, and they are not kind of sort of, you see how the Schwann cell kind of surrounds it like that, but doesn't kind of go around it, it just pretty much just insulates it. But it's not myelinated, you see, because it doesn't have the sheath all the way around, you know what I'm saying? So it will still conduct slowly, but it is going to be insulated from this one. So number 1 is insulated from 2, from 3, from 4, from 5. Okay, so oligodendrocytes do that in the brain, and Schwann cells do that in the periphery, okay? So they are still surrounded for insulation, and you can see the difference between the two. You got the Schwann cell here, or in the peripheral nervous system, or oligodendrocytes if it was the central nervous system. But here you also got the Schwann cell that kind of wraps around them, and unmyelinated, axons, because to kind of sort of insulate it from the other ones. Otherwise, they'll be busy cross-contaminating and stimulating each other. Okay. All right. So, because of the myelination and unmyelination, we have the matter in the brain and the spine that looks whitish because of the myelinated axons, which we're going to call white matter. Okay. So, consists of myelinated axons. If I say what does white matter consist of, you have to say myelinated axons, because only axons can be myelinated. Okay. But if you are talking about gray matter, you say unmyelinated structures, because there's plenty of them. Cell bodies don't get myelinated. Dendrites don't get myelinated. Some axons don't get myelinated. Okay? So many structures here. But for white matter, you have to say axons. Okay? Don't just say the myelinated stuff. You have to be specific about that, because it's only the axons that get myelinated. So it propagates action potentials much faster. Okay? Forms nerve tracts. So those will be the white parts of the nervous system, okay, the white matter, and the white parts of the thing, which I'm going to show in a little bit, of the spine, okay, and then the gray matter usually is inside, and it contains all the cell bodies and all the dendrites, okay, because those don't get myelinated. So anyway, so collection of neuron cell bodies and myelinated axons also, and forms the cortex. Remember, I was talking about the cortex of the brain, and I was saying it's only about 5 millimeters thick, and that's where 75% of neurons in the whole nervous system are located. So they are densely packed, okay? And it's the gray matter on top of the brain, okay? That covers the cerebral hemispheres. Okay, it's called the cortex, okay? Cerebral cortex. And then, of course, you have the nuclei, and they're called nuclei if they're in the brain. You see? The groups of cell bodies in the brain are called nuclei. And then, of course, ganglia, they're called in the peripheral nervous system, right? Okay, so you know what gray matter is, white matter is, all those types of neurons. And the function and characteristics of neurons, and the 2 basic cell types, and organization of the whole system. So, actions, of course, synapse with cell bodies to control them. As well as synapse with other parts, you know. So integration is pretty much a function of all these neurons, interneurons they're called, in the brain, connecting each other and processing information and then passing it on to the next part of the brain, which is called the next nuclei and so forth. And then once a response is formulated, then it's sent along the motor neurons to the effectors. So again, you can see the spinal cord. You got the dorsal and ventral root, and we'll do it next time. And that join to form the spinal nerve. Okay, and then the dorsal root is a ganglion, a cell body here that meet, and then so it will synapse with another neuron right there. So that's why you have the cell body there. And then of course you see the gray matter is in the middle, and then the white matter is on the outside. So the orange part is what is indicated. All right, and that's that. And then in the brain, of course, you've got the gray matter, which is the cortex. So this little thickness, that's the cortex, where about 3/4 of the neurons in the whole nervous system are packed in there. And then the inside is the white matter. Okay, and these are tracts that connect the two. This is called the corpus callosum. Okay, it's got up to the tune of 300 million axons crisscrossing from one side of the brain to the other side. Okay, 300 million, that's like the population of the United States, okay? Crossing from one side of the brain to the next. And you'll see it when we dissect the brain next time, okay? And it's so white. Thank you guys, I appreciate it. We'll see you next time. No, we'll see you now in 418. Is it 418? Over in the library building, so we can do this. We're still doing electrophysiology, right? Today is the other part. What?
You're not doing it.
I'm not here on Thursday, by the way, guys. I'll be my last tube of the day, of the season, and then Dr. Matos is going to be here. Okay? All right. Thank you, guys. I appreciate it. Whoa.